Subgroup meta-analysis and producing a forest plot. Meta-analysis is a statistical method that can be used to combine data extracted from several publications or several studies that are independent and separate, but all addresses the same scientific, medical, or clinical research question. And the objective of the meta-analysis is to provide or generate an overall estimate of the effect size when comparing between intervention and a control group. And the subgroup meta-analysis is sectioning the result based on a factor or a variable and performing separate meta-analysis for each subgroup. From the subgroup meta-analysis, a forest plot is generated with sections for each subgroup. Forest plot is a graph or a diagram that displays the summary of the subgroup meta-analysis. The forest plot consists of three parts. The first part is descriptive statistics for the subgroups meta-analysis, including the study effect size, the p-value, the 95% confidence interval, and the weight percentage. The second part is diagrammatic representation of the descriptive statistics, and the third part shows the calculated overall or pooled estimate effect size with p-value and heterogeneity test with an I-squared value. If you find this video useful, do subscribe to the channel and click on the notification bell, and give the video a like and share the video with others to benefit from the content, so the channel can grow and develop to include a wide scope and varieties of different statistical analysis in different statistical packages such as SPSS, Excel, Stata, Jamovi, R-Statistics, GraphPad, Prism, GPower, Python, GMP, and RevMan. Subgroup meta-analysis is a statistical method that is used in meta-analysis to examine and assess the effects of intervention or treatment within specific subgroup of population. So the studies in subgroup meta-analysis are sectioned or divided into two or more subgroups based on certain characteristics or a factor or a variable such as age, gender, disease severity, and so on. So the pooled effect size is calculated for each subgroup and compared to determine whether there is a difference between these subgroups in the estimated effect size. So the objective is to section the studies into two groups or more, and then perform meta-analysis for each group, then generate or calculate a pooled overall estimate of the effect size for groups, and then compare them to see if there is a difference and whether the difference is significant. Examples of factors for subgrouping in meta-analysis. One of the examples to perform subgroup meta-analysis is to include a variable that have a age of individual that are sectioned into children, adults, and elderly, and subgroup meta-analysis can be performed for the children, adult, and elderly, and then the result or the output is compared to find whether there is a difference between the three subgroups. Another characteristic or variable that can be used for subgrouping is the gender. The studies can be grouped based on the gender, whether there is a male or a females, and then meta-analysis can be performed for each gender. And another factor that can be used for sectioning the studies or the data can be the disease severity, such as the early stages or advanced stages. The data can be sectioned based on the level of the progression level of the disease stage. Another example is the body mass index, which can be used as a factor for sectioning studies based on three groups, such as underweight, normal, and overweight. Another example of subgroup meta-analysis is smoking status, in which there will be three groups or three subgroup meta-analysis, one for smoker, another for non-smoker, and then a third for a past smoker. And finally, an example of subgroup meta-analysis is the ethnicity or the race, in which the participant or recruited individual to the studies can be from different background, whether they are white, Asian, black, or others, and subgroup meta-analysis can be performed, and the outcome or the output for each subgroup can be generated, and the effect size for each subgroup can be calculated and compared. Here I had inserted a data from 12 clinical studies from 1 to 12 that are looking at the effect of specific intervention or treatment compared to control. So here I have an experimental group and a control group. In the experimental group, the event is counted and inserted here, and the total number of participants in that group is also included, and for the event occurring in a control group is also recorded with the total number of participants in that control group. So each study had sectioned the participant into either an experimental group or a control group, and the success or the happening of the event occurring in the experimental group is recorded, and the event occurring in the control group is also recorded. And if you look here, there is another variable, which is gender. So there are 6 studies that had only recruited males, and another 6 studies that had only recruited females. So subgroup meta-analysis can be performed based on the factor of the gender. So meta-analysis for the male group will be performed, and the estimate effect size will be generated, and the same for the females. And since here the outcome is binary, in which there are 2 levels, either the event happening or not happening, then binary meta-analysis will be performed, and specifically subgroup binary meta-analysis method will be used. Here I had placed a forest plot generated in R statistics for the previous data, and as shown here, the plot has 3 sections: descriptive statistics, diagrammatic representation of the descriptive statistics, and overall estimate of the effect size and heterogeneity test. And as shown here, the studies are sectioned based on gender for male and for females, and subgroup meta-analysis is performed for the male and then for the females. And in this third section here is the meta-analysis without subgrouping. And as shown here in the first section, for the subgroup meta-analysis for males, the experimental and the control, the events and the total is inserted with the weight percentage, which reflects the size of the sample. So larger samples will have larger or higher weight percentage, and that will have higher influence in the meta-analysis. And the estimate effect size is the odds ratio, as the outcome here is binary, and a fixed effect model is used, and the MH method is used. So here as shown that the odds ratio for each individual study is calculated and given here, and the pooled overall estimate of the effect size, which is the odds ratio, is calculated as given here with the 95% confidence interval. And here at this section the heterogeneity test is performed. Looking at the diagrammatic representation here, the blue squares represent the odds ratio for each specific study, calculated for each specific study. Variation in the size of the square reflects the weight percentage. Larger samples will have larger squares. The horizontal line represents a 95% confidence interval. The diamond shaped here structure represent the overall odds ratio. And the same thing here for the females. And if we look at the pooled overall odds ratio for the males is 19.3, and for females is 14.68. And if we look at this section here, this section shows the overall odds ratio without performing subgroup meta-analysis, and it is 16.03. So as shown here, there is a variation between males and females for this intervention or treatment when the effect size calculated is the odds ratio. So this method of subgroup meta-analysis will section the studies and will give more details and will calculate the overall odds ratio or effect size for each subgroup and then compare them. The forest plot also will generate heterogeneity test, which assess or examine the level or the degree of variability or heterogeneity between the results from those six independent studies. And an I-squared is calculated, which is equal to 0%, which shows that there is no heterogeneity, and the p-value has to be greater than 0.05. However, for the females, the heterogeneity was 36% as the I-squared was calculated, and the p-value is greater than 0.05. So there is a low level of heterogeneity between the females compared to the males. And also furthermore, the heterogeneity is calculated for the meta-analysis without subgrouping, as the I-squared was equal to 0% and the p-value greater than 0.05, and a test of subgroup difference is also given here. So this is the essence of doing and performing subgroup meta-analysis to section the data or result from studies based on a factor which is gender here. If you have a data and you want to analyze the data and you are looking forward for a training course or a training sessions, then I can arrange a one-to-one private tutoring session to help with the analysis. Whether the software to be used is SPSS, Excel, Stata, Jamovi, or Statistics, GraphPad, Prism, Revman, you will be welcomed.